Our first lab examines the macroscopic and microscopic anatomy of the endocrine system. You may recall from AMP1 that this organ system is responsible for homeostasis. Unlike the nervous system, however, the endocrine system response times are relatively slow, but the effects are longer lasting. A little Professor Klein note with histology. If it looks different, that's because it is different. We will repeat that all semester. This is because every tissue type is unique and stains differently. So remember that. If it looks different, it is different. So what defines an endocrine gland? Well, these organs contain no ducts and therefore release their secretions into a body fluid. These fluids include blood, lymph, and even interstitial fluid. The secretion, a hormone, will travel through this liquid and activate another target cell. For example, insulin may be released from the pancreas, travel through the blood, and stimulate adipocytes to store glucose as fat. This laboratory will discuss the major endocrine glands, the pituitary, adrenal, thyroid, and parathyroid glands. The remaining endocrine glands will be discussed in their organ system. The pituitary gland. This organ is controlled by the hypothalamus by way of the infundibulum. However, the two structures in the pituitary come from different embryonic origins, which affects the control mechanism by the hypothalamus. The anterior pituitary, or adenohypophysis, is derived from a different embryonic tissue layer as the hypothalamus. As a result, these two organs are connected by a special portal system of blood vessels in the infundibulum. The hypothalamus then controls target cells of the anterior pituitary by way of releasing hormones. There are many hypothalamic releasing hormones and anterior pituitary hormones. These include TSH, FSH, LH, GH, and prolactin, or thyroid stimulating hormone, follicle stimulating hormone, luteinizing hormone, growth hormone, and prolactin. The posterior pituitary, or neurohypophysis, is derived from the same embryonic tissue as the hypothalamus, and as a result, they're connected directly through neurons. In fact, the soma of these neurons lies within the hypothalamus. Axons run through the infundibulum, and the axon terminals are in the posterior pituitary. The hormones of this structure are released directly from the posterior pituitary. There are only a couple of hormones released here, oxytocin and vasopressin, which is also called antidiuretic hormone. These hormones are made in the hypothalamus but released from the posterior pituitary. Regarding the histology, we easily see the difference between an adenohypophysis and the neurohypophysis. The adenohypophysis is darkly stained because it's far more cellular. Recall, this is a different tissue type and full of actively metabolizing and secreting cells. Whereas the neurohypophysis is less cellular, containing unmyelinated axons and secretory axon terminals, along with some glial cells. In this second slide, we see the various cell types of the adenohypophysis, or anterior pituitary. These three cell types pick up different stains and look a little different as the cytoplasm is stained differently. Remember our rule? If it looks different, that's because it is different. Acidophils appear pink and secrete growth hormone and prolactin. Basophils appear purple and secrete thyroid-stimulating hormone, adrenocorticotropic hormone, follicle-stimulating hormone, or luteinizing hormone. So those would be TSH, ACTH, FSH, or LH. Lastly, the supportive cells, chromophobes, are the lightest in color as they don't pick up the stains very well. Remember, if it looks different, that's because it is different. The adrenal glands. As their name implies, they lie on top of the kidneys. Each gland is organized into two zones, the outer cortex and inner medulla. The cell types in these regions vary, and therefore the types and the functions are different. The cortex zones from superficial to deep are the zona glomerulosa, which secretes aldosterone, the zona fasciculata, which secretes cortisol, and the zona reticularis, which secretes androgens. In this labeled photomicrograph, we once again see those three layers within the cortex region. Note how they do move from superficial to deep, and they are visually slightly different from each other. The thyroid gland lies just anterior to the trachea and even houses a different endocrine gland. This gland secretes two forms of thyroid hormone as well as calcitonin. Again, we see that trend, that different cells and tissues look different. The thyroid gland is organized into three-dimensional follicles formed by follicle cells. These cells are responsible for converting thyroglobulin in that pink staining colloid into active triiodothyronine or T3 and thyroxine T4. Between the follicles lie clusters of parafollicular cells which secrete calcitonin. As mentioned on the previous slide, the parathyroid gland is found within the thyroid gland. This odd organ is organized into a series of secretory clusters with different cell types. The larger and lighter oxyphil cells are a mystery in terms of function. The smaller and darker staining chief cells secrete parathyroid hormone. Note the scattered clusters of adipocytes. And by the way, calcitonin and parathyroid hormone should sound familiar from ANP1 and calcium homeostasis in the skeletal system. Now, navigating the laboratory chapter and using the website to complete this exercise should be more efficient now that you've had an introduction to the organs, cells, and hormones. Once you complete the laboratory activities with the website, make sure you complete the review questions. Always, always, always use your original words when explaining concepts. Don't just copy the lab manual. If you have any questions, send me an email.